Hi, my name's Nuria. I'm going to tell you briefly my poster title, Digging into the Molecular Differences Among Conditions by the Integration of Spatial Transcriptomics Samples Beyond Gene Plotting. Don't hesitate to contact me if you have any further questions. RNA sequencing allows us to uncover general molecular differences. However, this technique has not enough depth to identify more subtle differences in concrete populations. Spatial transcriptomics approaches allow to sequence mRNA in situ in spots from a sample slide. Its strong point is the gene spatial location. In this recent scenario, we had the following question. Can we compare different conditions to get disease-associated signatures in cell populations using spatial transcriptomics? We applied this problem inside the context of autoimmune thyroid diseases, Hashimoto's thyroiditis, and Graves' disease, compared to controls. We used 10x genomics technology for that aim. First of all, we compared in silico clustering versus pathology-based. In the second step, we used Harmony 2 sample integration and sorted pipeline to evaluate differences within thyrocytes, connective tissue, and vessels among conditions. In the third step, we validate these results using a public single cell from Hashimoto's thyroiditis samples, and finally we validated by immune staining, but these data were not shown. Regarding the comparison between in silico clusterization versus pathology-based one, you can see that both techniques were quite similar. However, due to cluster purity following histopathological criteria, we decided to use this method to isolate the main cell population and for further analysis. When we performed a sample integration using Harmony to correct batch effects, we saw a correct integration of samples. Data were not shown. Pathology-based groups properly distributed, and also conditions. After that, we took main markers to evaluate the groups as we can see in the dot plot. Then we isolated vessels, thyrocytes, and connective tissue groups to evaluate independently their differences among conditions. Regarding vessels, differential expression analysis between Hashimoto's thyroiditis and controls and Graves' diseases and control showed an upregulation of some angiogenic markers in both autoimmune thyroid diseases. Remarkably, just in the case of Hashimoto's thyroiditis, we saw an upregulation of ACKR1. When we saw both angiogenic markers, we saw an upregulation within Graves' disease samples, as you can see in the images. Regarding thyrocytes, we observed a cluster with a remarkable presence of autoimmune thyroid diseases spots. When we performed a differential expression analysis of this cluster against the others, we observed an elevation of damage-associated genes, antigen-presenting markers, and even more, a low thyrocyte differentiation score or TDS, but these data were not shown. Spatial representation of this TDS reveals that lower score is close to immune infiltration as long as antigen-presenting markers, but these data were not presented neither. When we validated this presumption in single-cell data, reclassing of thyrocytes showed two populations, one healthier with higher TDS, another one damaged with lower TDS, an increasing of antigen-presenting genes, and also an increased response to stress. From the clustering of the connective tissue, we selected the top 40 markers of the purest cluster to perform a hierarchical clustering. We highlighted these two signatures to be associated to concrete fibroblast subpopulations in autoimmune thyroid diseases, myofibroblasts, and immune-associated fibroblasts. Spatial representation and density plots revealed that there is a higher expression of myofibroblast signature in Graves' disease, whereas there is a higher representation of immune-associated signature in Hashimoto's thyroiditis. We confirmed the existence of these two subpopulations in single-cell analysis. To conclude, spatial transcriptomics allow us to integrate samples and to infer molecular distinctions in a cellular level among different conditions. In autoimmune thyroid diseases, we unraveled subpopulations of thyrocytes, fibroblasts, and angiogenic processes concerning Hashimoto's thyroiditis and Graves' disease. Thank you for your attention. Hope you enjoy this poster.